everybody, this is Miss Nicole with Science Bytes at Bloomfield Public Library, and I'm really excited about the project I have for us today. Sometimes projects just sort of come to you, and this is one of those examples. USA Science and Engineering Festival partnered with a company called Illumina, and Illumina is a genomics company. They work with genes and they study DNA. That is why we are doing DNA extraction from strawberries. And all of you should have seen some of our advertisements. We do have kits available at both of our Bloomfield Public Library locations. The outside of the kits look a little like this. And in those kits are all the materials, except for a strawberry, that you will need for this project. And we still have some at the library, so if you didn't get a chance to pick up your supplies, by all means, give us a call for a curbside pickup. DNA is actually a shorthand way of saying deoxyribonucleic acid. And that's a mouthful. DNA, actually in our bodies, it looks like sort of a twisted ladder. It's two strands that are connected by little rungs. But it's so tiny that we would need a very high-powered microscope to see that DNA. So we won't be able to do that visually with our eyes. We won't see those ladders. But all of the information that makes us who we are is contained in cells inside our body. Think of a cell like an egg. And in order to get to what makes us who we are, we have to crack that egg open. DNA is in every living form on this planet. And all living forms share some DNA in common. So if you think about all the things that are alive in the world around you, trees, fish, lizards, we as human beings share some DNA in common with all of those life forms. And interestingly, we actually share 99% or about 99% of our DNA with a chimpanzee. And you would think that would make us very similar to chimpanzees, but that 1% makes all the difference. If you look at the two of us side by side, chimpanzees seem very different than human beings, but inside we share almost 99% of the same DNA with a chimpanzee. Now, the only thing I didn't supply you with is a strawberry. And you can use fresh strawberries or you can use frozen strawberries. I sort of have a plate of both here. Here are my fresh ones and here are my frozen ones. And the frozen ones might actually be a little easier to work with because they're a little squishier. I don't know if you can see, but they're a little squishier. They might be easier to mush because we're gonna have to mush these strawberries. What I did with my fresh strawberries is I left them on the counter for a few days so they would ripen a little more. So they're also a little bit softer than they were when I first had them from the food store and put them in the fridge. Notice that one of the items in your supplies is a tube that's labeled 70% ethanol. That's a kind of alcohol. And this is going to be one of the items we work with. And I am going to recommend get a glass and fill it with some ice and then just push that test tube right into the ice. And the reason that we want to chill that ethanol is that DNA actually degrades or starts to disintegrate or fall apart in warmer solutions. So if you keep things at room temperature, you'll still be able to see it with room temperature ethanol, but chilling it helps protect that DNA just a little bit longer. It helps preserve it and make it nice and visible for us. So cooling it is just a way to protect that DNA and let us play with it a little longer while we're doing our project. Now for both of these chemicals, the ethanol and the extraction buffer, like with any science project, you want to be careful. They're not dangerous, but if you did accidentally get something in your eye, you would want to flush out your eyes right away. And if it was really bothering you, you would talk to a doctor. But there is no reason that we would get this in our eye as long as we're being careful, because it's not going to splash around. And we just want to make sure that as we're doing this project, we don't work with these and then touch our eyes. So just make sure that you're being careful And I did also include in everybody's bag some gloves. These are not necessary, but if it makes you feel more comfortable to use gloves, you can use gloves. Now the first thing you're going to do is look for this smaller Ziploc bag. If for some reason you misplaced it or something happened to it, you can absolutely use your own Ziploc sandwich bag from home, and it's going to do exactly the same thing. The Ziploc is the important part. We want to make sure we can seal the bag. I'm going to take 2 of my fresh strawberries and place them right inside this bag. And I think I'll also use this one. I got 2 strawberries in here. So I'm going to try and squeeze out some of that extra air. Sometimes if you fold the bag like this, you squeeze out that extra air. And now I'm going to zip the little Ziploc at the top. So now I've got these 2 strawberries that are sealed in my bag. Now, you probably want to have some sort of placemat down just in case you have some sort of an accident where the Ziploc, the top of the bag isn't sealed perfectly and something squishes out the top. But once you've got it on your surface, you're just going to start to literally press. And every time you feel a little lump of strawberry, you're going to gently smush it. That's my technical scientific term. You're going to smush it and break it up. And what we're doing right now, as I mentioned before, is we are actually cracking open all those cells inside the strawberry and helping to release the DNA. We're going to do this for a couple of minutes because we want to break up as much as possible. Now is the time for us to use our extraction buffer. And for us, it's the solution that looks a little bit pink, but it's also labeled extraction buffer. And these caps actually screw off. You just untwist them. So I'm gonna open my bag first. So you're gonna make sure you pull those strawberries away from the top of your bag, because we're gonna open the Ziploc bag like that. And now we're gonna unscrew the top of our extraction buffer, and we're gonna pour that into our bag and reseal it. So here I go. And again, you wanna try and not have a lot of air in that bag. So I usually fold it over a couple of times and then seal it like this. And now once you unfold it, there isn't a lot of extra air in there. And you're going to very gently mix that extraction buffer with the strawberry juicing that we've already sort of broken up. You don't need to do a lot of heavy mixing, just gently press it. And that is helping that DNA to come out of the cells even further. Now, this next step of the project is probably the trickiest. And it's the trickiest because it doesn't always go exactly as you planned. So for our next step, we're actually going to strain our strawberry juice through the filter that was included in your kits. And this filter is made of paper. So you can imagine what probably happens once we add liquid inside this paper filter. This is going to start to break apart. It is only paper. If you find that your filter starts to break up and you get a little leak, try and just cover that leak with your hand, because we are going to try and just squeeze that strawberry juice through this filter. And we don't want the chunks. We don't want any little bits of strawberry, just the pure juice, the juice in the extraction buffer. So if you spring a leak, just try and fold it up a little and cover it with your hand and continue to strain through the rest of the filter. Now you can see from our previous step that we've got a good bit of strawberry here. We do not need to put all of this into the filter. We're going to pour just enough of it to strain through, so maybe half of this. So you do not have to dump this entire amount of strawberry into that filter. Here we go. I'm going to open up that paper filter, pour in my strawberries, and I'm going to gently squeeze and strain it through. We want just the juice. You can see I'm not getting all of it in the cup, and that's okay. That's why we also use a placemat. Now, once you're done straining your strawberry mixture, you should have a clear solution in here. It should be just strawberry juice. If your filter accidentally broke open and you got a few pieces of strawberry in here, little clumps of unsmushed strawberry, you don't have to start all over from scratch. Go ahead and get a little spoon or use your fingers and just pull those lumps of strawberry out. We just want the strained strawberry juice here. This was the extraction buffer plus our strawberry juice. So we are now going to take the ethanol, which we were chilling in our glass, unscrew the cap, and we're just going to gently pour it right in to our strawberry mixture. And almost right away, you can see some strange little white filmy things forming on the surface. So here you can see a little better the two layers that we now have in our cup. And if we look at the top, we see those strands of DNA. Now, you did also get a wooden stick inside your kits, and you can actually gently move that DNA around. See that little glob? That weird, slimy, white, strandy stuff is our strawberry DNA right there. Now there's one additional thing we can actually do with this project. Everybody got these little tubes with lids inside their kits and a piece of string. Now the folks at Illumina actually thought it would be pretty cool if you could put some of that strawberry DNA into this capsule, seal it up, and put some string through it so that you could wear it as a necklace even if you wanted, or just hang it on your wall. I will say that if you do that, make sure that for the most part you keep it chilled in the refrigerator and you only pull it out when you want to look at it, or for short spans of time. Or if you're okay with it degrading and you want to wear it to school for one day, that's okay too. But just know that if it's not chilled, it will degrade faster. So everybody also got one of these nifty little pipettes in their kits. And a pipette is a fancy way of saying an eyedropper. You might be more familiar with that term. So when we squeeze this bulb at the top closed and put this in something and then let go, it's gonna slowly suck that liquid up into this tube. And that's what we're going to do. We're gonna actually suck the DNA up into this and then transfer it into our little capsule. So if you watch me here, we're gonna slowly, we want just the top part, not the bottom, so you're only putting it in the top. And you can see how I'm getting the DNA, right? And now, we're gonna just put it right into our capsule. And there we go. And now you're gonna seal it up nice and tight. Wait till you hear that click. And now you can just take the thread that came with your kit. I'm gonna find the end of my thread. And there's a little gap right where the capsule closed, and you can feed this through that gap, and like that. And now you can have a nifty way to carry it. You can wear it as a necklace. It is up to you. But now this capsule contains strawberry DNA. I do have some extra supplies of the alcohol and the extraction buffer that was given to me by Illumina. So if something went wrong, or if you were dying to do a second version of the experiment, just call us at the Prosser location of the library and bring me your empty little test tubes with their lids. You want to make sure you have the lids with them, the little screw lids, and I can give you some extra solution. Hope you had fun with this one, and I will see you at our next Science Bites. Bye, everybody.